I think Jocelyn did a really amazing job explaining Visium and everything we can do with Visium. And we looked at what everybody was saying that is attending, and Visium is definitely the one data set that most people are familiar with. Visium HD is a new technology, I would say, from 10x Genomics. It's kind of like Visium on steroids. It's pretty cool. It has a much higher resolution. I will go into this in a bit. So I want to show how you can analyze Visium HD with Giotto Suite at the highest bin resolution of 2 micron, using our very flexible tiling and aggregation strategies that are directly available in our toolbox. But a very similar strategy can be used on any other similar technologies like SteerSeq, SeqScope, OpenST, PixelSeq. There's many other spatial sequencing-based technologies that operate at the subcellular level, because that's really where we're getting at right now. Like Visium is great, but these spots are like 55 microns, so they're quite big. There's a lot of different cells within a single spot, and this is why you had to do deconvolution and think about what are the different cells that are available. So it makes interpretation a little bit hard, and I think that's hopefully what we can change. Well, when people are kind of like moving into these new technologies. Um, so I am kind of like assuming that you know all the steps that Jocelyn just described, including PCA, UMAP clustering. So we're going a little bit faster through this tutorial than we did with the previous one. But if you need, you can always go back to part one and part two, because like some of these default steps are very similar between any data analysis step. One thing I would like to say, and it's a little bit, people kind of like think, hey, we figured it all out because we can now read out genome-wide information at a 2 micron bin, or with SteerSeq, it's even a 500 nanometer bin. There are still some issues. Capture efficiency can be quite low sometimes. There's also lateral diffusion, where your transcript might kind of like diffuse outside, and maybe it's a little bit debate whether that's 1 micron or maybe even a few 5, 10 microns, and what that exactly means. So there's definitely also room for other technologies. And for example, I think George in the afternoon is going to present Xenium, which is based on in situ hybridization. And if you remember what I said yesterday, like what in situ hybridization can do is actually capture the single transcripts at a super high resolution. Nevertheless, Visium HD and these other type of tools are really interesting if you have like no idea, or you just want to have like an unbiased view about what is expressed and what we can learn. So Visium HD is very similar as Visium, as I said before, except that it's not spots, but like it's a continuous field, and they're at 2 micron bins. You get these spatial barcodes, and they can capture like multiple transcripts. It's not just a single transcript; it's multiple transcripts, and sometimes the same transcript can be captured multiple times, meaning there are multiple UMIs for the same gene. So I think that's all pretty amazing. And that also means that your data sets all of a sudden are now going to be pretty big. So what 10x already provides is their highest resolution is 2 micron, but they do already aggregate that data in 8 by 8 micron bins, but also the 16 by 16 micron bins as a kind of like an aggregated data set. All right. And I'll come back to that later on, because we use a slightly different and I guess more flexible strategy, although you can definitely load in these data as well. So for this specific example, we are going to use the colorectal cancer sample. It is pretty big, and that's actually why I actually subsampled this data set and I made it available on Zenodo. But just to get an idea about how the data set or the data structure looks like, it's definitely a little bit different compared to Visium. And here is the format of the folders of the Visium HD dataset, where, as I told you before, 10x already provides previously aggregated data, whether this is at the 2 micron, which is not really aggregated, that's just the highest resolution, but then you have the 8 micron and the 60 micron. So if you unzip that one, you can actually see that there has a spatial subfolder, and within a spatial subfolder, the key, or one of the key files that we need, is the tissue positions per get file, because that's the file that tells you the exact pixel coordinates for each individual pixel within that Visium HD And then very similar as what we've seen with other 10x formats, you have the raw feature or the filtered feature matrix, and that is basically your matrix format. So that's where all your genes are being detected, or you can identify how many times your gene is detected. All right, so let's keep moving on. So as I said before, I did create a mini Visium dataset, and that is needed because the dataset is actually quite large. And we'll talk about a little bit about what we can do to alleviate that concern. And there's many different ways. I think it's the best way right now, if you really want to analyze a big Visium HD dataset or any other really big spatial dataset, is to try and look for a really fancy local machine if you have enough money, or you use a server or an AWS or whatever else you have available, because otherwise you might run into memory issues. And we'll tell a little bit about how we are trying to alleviate some of these memory issues. And at the end, I'm also going to give the floor to Eddie, who's going to discuss a little bit about his like really promising work to even work with like these really large data sets on your local machine. But that's for the end of the session. All right, so what you will need to do is you will need to start a library. You load Giotto if you have not done that. Again, I would recommend you to restart your R session if you have been following the previous tutorial, just to make it a little bit easier. You can set up a couple of paths, and then what we would like to do is just download this smaller Visium HD dataset that I created, which you can find on this Zenodo link. And the thing, if you kind of like unzip it, what you will find is that I just store the tissue positions Parquet file in this raw feature matrix, because that's the only thing we're going to use. All right, so how do you load in Visium HD data in Giotto? There are definitely two ways. The easiest way is to use our convenience function, but I'm not going to use that. I'm actually going to show how you can do this manually, and in this way you can actually do it with any other type of technology. So if you, for example, have SeqScope data, or you have OpenST data, or Nova ST data, or Pixel-seq data, so anything similar to Visium HD data, you can follow a similar-ish strategy. You might have to change a little bit, but it will work. All right, now I'm going to switch to my other screen. All right, now we are in business. All right. So we're back here. So I already loaded or kind of like downloaded files. So I just told you that I want to read in the data manually. So the first thing I do is I read in the raw expression data, right? So that raw expression data is actually very similar to any other type of data. It's just the gene by pixel type of data, except what we don't have for each pixel. And can maybe just show you really quickly how that looks like. So these are the individual genes, and then the column names are these pixels. But what we don't have right now is we don't know what the spatial coordinates are of these pixels. That information is stored in tissue positions. So we can read it in this tissue positions. And what you see here is for each barcode, you can now see the tissue, the array, and then also the pixel information here and here, the column. All right. So what we hope to do is we want to combine the expression information and the tissue information. If we do this, and then we get this. So now we have what I call here a matrix.tile.dt, where we have for each ij, so that's this array information that Visium HD has. We know exactly what is being expressed and which gene it is and what pixel it's associated with. We can combine it with the tissue information and load that one in. And now we also have the XY coordinates. So we have for each pixel, we have the XY coordinates, and we know which genes are detected and also how often it's detected. And that is what I was telling you before. That is the UMI. Like if a gene in the same pixel, if there are like 2 transcripts for a gene, it will have a count of 2 because you will have 2 UMIs. So this is really kind of like the raw data that Giotto can use to represent your what we call Giotto points, and I will go back to this in a minute. So it's simply meaning which genes are detected or which transcripts are detected at which location in the XY coordinates. That is your Giotto points. Now, what we also need is we need to have a spatial unit. And if you remember what I said in the introduction yesterday, is that a spatial unit is what we define as like, what are you using to kind of like understand the spatial resolution of your data? And we don't necessarily make use of the pre-aggregated 8 or 16 micron format, but we can generate any type of tiling or whatever you would like to use. And here I would like to show this, that we can create hexagons. And these hexagons can have different sizes. And using these different sizes, you can make it easier or harder to analyze your data. First, we're going to create these Giotto points from this data that I stored before. So this is the expression position data. So that is the data that gives you information about each transcript and where it is found and how many UMIs it has. That is now stored as Giotto points. So here you see the Giotto points and how what is represented. We represent it as a spat vector. It's not necessarily needed to understand what it means, but it is a way to store your data in a very neatly organized spatial format for spatial data. All right, but okay, so we have our features that we represented by Giotto points, but now we're going to create Giotto polygons. As I mentioned before, since we operate at 2 micron, we will have to aggregate these 2 micron bins in one way or the other. And so we can use our very flexible tiling aggregation function. And we can really represent any type of tiling aggregation, right? In reality, it's just these tiles are also just a bunch of polygons that have a very specific symmetrical format and that cover your whole tissue area. And here I'm just giving you this idea, like these are hexagon tiles and how they kind of like can cover everything. And you can decide how big you want these tiles to be, where you want them to be at the single, or you want them to be really big or really small. It doesn't have to be hexagons, right? We already have like preset ways of working with squares. This is the same way as what 10x does. You can use triangles, but I'm here using the hexagons. You can also use any other type of format that has this kind of like symmetrical pattern. Or if you happen to have polygon information, let's say you have an IF, like immunofluorescence kind of like data on top of your tissue, and you can identify individual cells, you could also use that and just like design your polygons. And this is something that I think we will cover a little bit more in the Xenium tutorial, where we can identify, where we have that information, we can identify these individual cells and their polygons, their shapes. All right. But here we're going to create this hex bin, and here you see the hex bin. So this is what is basically what we created. It's based on the extent of the Giotto points. So the Giotto points, again, as a reminder, these are the information about where the transcripts are being expressed. So they have a specific extent. So that is an XY dimension where everything is expressed. And then we create these hexagons that will cover all the different transcripts. And you will see in a bit how it actually looks like when you combine both of them. Right? So here now we are going to create our Giotto object. So we're going to create a subcellular Giotto object because we work with subcellular data. And there are 2 essential parts of the subcellular data. It's the Giotto points that we created. That is where you will have your feature information, and the Giotto points, this is where you will provide your spatial unit information. In this case, it's our hex bins. I like to create my Giotto instructions, and I provide it with a saving directory. This is where I'm going to save all my plots. I'm going to say save plot is true, show plot is false, return plot is false. Not everybody likes doing this, but I prefer doing it this way, because what you do, you save all your plots directly to a folder rather than trying to show it or return it to the console. Because if you try to show it in RStudio, that actually takes so long, and especially with like really big data sets, all of a sudden you're just waiting like minutes just to render pictures, while if you just save it away, it will be converted into some kind of like PNG or JPEG or whatever it is, and you can open it almost instantaneously. So we created this object, the subcellular object. One thing I'm adding are the spatial centroid locations, because a lot of what we do in Giotto kind of like depends on the centroids. In this case, the centroids is usually for each spatial unit is a centroid, and that is simply the middle, in this case, of your hexagons, right? That's also something we add to the Giotto object. All right. So now I just want to visualize the data so you have an idea what exactly we created. One thing we need to set, and I will show it also in the exact function, is we need to set in the expands counts is true, because we want to expand the counts column, and we also and we want to set jitter to some form of degree. And so why is this? And I'm probably repeating myself. So what we have done so far is we have generated hexagons. This is one hexagon. And then we have converted our 2-micron expression tiles into these Giotto points. But in reality, each 2 by 2 micron tile can have multiple genes, but they all share the same exact XY coordinates, right? Because that's just the XY coordinate of that pixel where those transcripts landed and where it was kind of like getting the spatial barcode. So you need to kind of like expand the counts, and you need to also give some jitter so that this one dot will become like many dots. So in this one dot, in this specific pixel, what you would find is that that pixel had like 3 different genes, this red, green, and blue. And each gene had also like multiple UMIs. This one had like 3 UMIs, the green one, this had 2, the blue, and the red one also had 3. And that's one way you can visualize all the individual transcripts, giving a little bit of jitter and expand the counts. That's just for visualization purposes. When we do anything downstream, we don't necessarily want to change the XY locations. We want to keep them as closely to their original location. All right, so we can run this. And what we use to plot subcellular data is the function spat in situ plot points. It's analogous to some extent of SpatPlot2D, which we have used a lot for Visium data, but this is more for if you have subcellular data. So we're simply telling it, like, show me Visium HD. I'm going to give it a set of features. You need to provide which features. I'm taking that from the feature data. So this is the gene metadata. I'm telling it, just giving me like 10 to 20. Show me the legend. And then I need to specify which spatial unit you would like to visualize. And you might say, hey, why is that? Because we can actually add multiple spatial units, as I will show you later today. And changing every spatial unit, you can do different type of analysis, and you can quickly go back and forth between different spatial units in your Giotto object. There's a bunch of other parameters you can figure out how to make your data looking a little bit better. And I'm just going to show you this. So this is a little bit what I was saying before, and probably this is the best way to understand what we have done. So we have loaded in the individual feature information, and that is being visualized. We're visualizing 10 different genes, or maybe a little bit more. Each of them have a different color. And now we also created this polygon. So this is a spatial unit that right now we're interested in, and we would like to use downstream. At this point, we haven't aggregated anything yet. It's just like we created the feature information, the Giotto points, and we created the spatial unit information, the hexagons. Please, you can also like interrupt me if anything is unclear or if you want me to expand on anything. All right. One thing I just want to briefly highlight is, so we plot most of our plots with ggplot, but if you have a lot of different points, which you might have with this type of data, that might take a really long time, especially if you're trying to render it to your RStudio session or whatever you use. So if you set here plot method to scatter more or scatter most, then you are actually going to create a PNG. It's going to convert everything already in the back end. I'm going to see if I can actually zoom in. This is doable. And oh, what is happening? Let me see. Okay, it's a little bit hard. I think if you are in your own local machine, you can try to zoom in, and then you will see it yourself. Like you do lose a little bit of in resolution when you go to and you use these tools like scatter more and scatter most, because you're basically converting your individual dots to more pixelated representation. All right, so now we can process our Giotto objects. And so this is now slightly different than what you've seen with Visium, because we do need to calculate the overlap between the points that we created and the polygons, right? And the polygons in this case are our hexagons. So we do want to create this aggregation so that we can create a matrix that we can work with. And so the first thing we need to do is we say, hey, let's calculate the overlap. So that's the overlap between the points, the genes, and the hexagons, the spatial information, which is in our case now hexagon 400. That's that. We can do then overlap to matrix. So this is now going to make an actual matrix from that overlap information. And it's automatically going to set an active spatial unit. And this is why this is important to understand, because your Giotto object can have multiple spatial units if you have multiple different polygons, like multiple different hexagons or whatever it is. You can set that spatial unit to different specifics. These are the default processing steps. I'm not going to go into detail. This is to some extent very similar to what you have done previously with Visium data. It's just like filtering. You don't filter based on the spots right now, but it's based on the hexagons, the hexagon with a 400 distance. You can normalize, you can add some statistics. Now we can start visualizing it. We can create that one. So this is just visualizing using the old SpatPlot 2D. And this is what I was telling you before. The SpatPlot 2D uses your centroids to visualize it. So you see something like this. But if you use the spat in situ plot points, then you can actually visualize the real, this is what you see here, the actual polygons that you use. So these are these hexagons that you can, in this case, look at the number of detected features in each of these hexagons. And they can be quite high, up to 3,000 different features. So that is pretty impressive for Visium HD. All right. So same way as before, we can do some clustering analysis. We can calculate the highly variable genes. We can do PCA. We can do scree plot. We can look at the number of interesting principal components that we would like to highlight. And again, I'm going over this pretty quickly because that's all been covered in the previous session about Visium. We can plot the PCA. We can visualize the PCA. We can then run the UMAP. We can plot the UMAP. We can perform we can create a nearest network based on the principal component space. In his case, I'm selecting 14 principal components. And we can do latent clustering. So this is graph partitioning based. There's also other tools that you can use. I'm using the one that is like embedded in R. It's two latent cluster iGraph. I think others might have used two latent cluster, but this one doesn't require Python if you have any Python issues. You can plot that UMAP that's already represented in this one. So now you see all the individual hexagons and the clusters that they fall into. Now you want to plot it back and overlay it with the original hexagons. And that's basically what you see. And you might be happy, you might not be happy. This is just like you have done this right now at hexagon 400, which is a little bit big. It's not, and maybe this is what you're seeing is more really an aggregation of multiple cells. So let's try to go a little bit lower and see what we can do. And that is a little bit one strategy you can use with these really big data sets is you can start with a higher aggregation, with a higher or a lower resolution, aggregate everything, identify interesting regions, and maybe like zoom in or try to understand what's happening. So just to show you that we can now add another hex bin, and now we're going to hex bin 100. And so we're going to do this, and we're going to do some additional analysis and see if we find anything that looks better. So the standard subcellular pipeline, as I described before, is like you create a new spatial unit, this layer, this hex bin 100. You add the spatial units to a Giotto object. You calculate the centroids. You compute that overlap with the transcripts and these hexagons, the new hexagons that you created, and you convert that overlap data into a gene by polygon matrix, which you can then put back into the default kind of like Giotto pipelines and everything you can learn. So we can run all of this. We can set the active spatial unit now to hex 100 because we have two different spatial units. So, and you can also set this manually, by the way. Each function, you can say, hey, I want to visualize hex 100, I want hex 400. But if you set the active spatial unit, you don't really have to think about it, so it saves you some time. Although I do see that actually I do explicitly also mention it here. All right, let's get this one. So, all right, so now you already see we're plotting again a couple of genes that we selected, but now as you can see, and you need to kind of like zoom in on your own, that the hexagons are definitely a lot smaller than the previous ones. So we are now already operating at a much higher resolved spatial resolution. We're going to do the calculate overlap, the overlap to matrix, everything that we have done in the previous, for the previous hexagon layer. Now we're going to do this for the hex bin 100 spatial polygons. All right, so that's going to take some time. So this is quite fast right now, simply because we are using a smaller data set. This might give you some issues when you try to do this with the whole data set. And I think this is a little bit what Eddie will also talk about, that we have thought about some ways that we can alleviate some of these problems in the very near future. All right, each is potentially important to understand, but like each spatial unit, and we have like at least two right now. We have one hex 100 and one hex 400, also has its own cell metadata or phenome metadata, and has its feature metadata. So you can do analysis on different spatial units and keep the information for all of it, and potentially think about how to combine all of that information in the future. I want to kind of like show some potentially more interesting. So we're just going to go through everything. We're going to do the calculate HIP again, the HPF, highly variable features. We do run PCA, we get that PCA. We're going to run the UMAP. Now we're already getting a little bit better or more interesting results from the UMAP. That's that. We're going to create a nearest network. So all of this is very repetitive, right? I'm just doing the same thing as I did for the hex bin 400. Now I'm doing this for a high resolution hex bin 100. We can plot that, but it's always more interesting when you can look at the SPAT and C2 plot points. So it's going to tell you that everything has been plotted. And now you can see this one. And this one, I think, is already a lot more interesting. You can already see that it has a lot more information compared to the previous one. And that's simply because you're working at a much lower resolution, where you capture probably cell types that are very similar and more like local changes rather than these big tissue organizations that we were looking at previously. So maybe say, hey, this looks pretty cool, and you can look at the image, which I didn't load right now. I kind of like see, does this somehow correlate, or is it similar to the H&E that is associated with this? And maybe I should have added it, because then you would have seen it. You say, hey, let's look at the spatial expression patterns. And again, I think Jocelyn already went over this, so we can go pretty fast. So we're going to use BinSpect again, although you can definitely also use a bunch of other tools to identify spatial variable genes. So these are just genes with a spatial coherent expression profile. We're going to restrict our analysis to only the highly variable genes. Highly variable genes don't necessarily mean spatially variable genes, but usually there is some overlap between a highly variable gene and a gene that shows spatial coherent expression pattern. The creation of the spatial network, and this is potentially important for BinSpect at least, is important. Depending on the neighbors that you use, you might get very different results downstream, although it doesn't change dramatically, but you do try to, I mean, the whole idea about BinSpect is that it tries to see if your neighboring genes also have a super high expression. So depending on if you only look at the 4 nearest neighbors, if you use a KNN network, so a K nearest neighbor network, in spatial context, or you use 10 or 20 or 30, might change the idea about what exactly that means. All right, so we can visualize these spatial genes per expression bin. I'm going to go a little bit faster just to give people enough time. What I like to do with the output of BinSpect, and this is this rank test, I like to separate it based on expression levels, simply because we see that different genes with different high expression levels, or number of high expressing genes, kind of like have very different profiles, and it's hard to kind of like disentangle the two of them. So you can visualize this one. So these are like some genes with where you see like very small expression patterns. This is from the lowest group, from the smallest group. But then you can, for example, do this one, and you start to see some genes with some interesting patterns. But as you can see, this is for just individual genes. And I think the coolest thing is really to look at spatial co-expression modules. Again, this is something that Jocelyn showed before, so we're going to go through this real quickly. So in my case, I like to pick a set of genes that have a very strong spatial coherent expression pattern, and you can set the adjusted p-value to something low, and you have a subset of genes, and then you visualize your co-expression heat map, right? And that gives you a pretty good idea like how many clusters you visually see, and you can kind of like set your K, the number of clusters you want to try to identify next based on that visual inspection. You might have to play a little bit with the number, but that's not too difficult. So you can identify these genes, you can create meta features. So in this case, what we did is we identify we're going to combine the genes that are in this co-expression modules. So if this one is identified, for example, there's a bunch of genes, we can put them in a meta feature, right? So that's a meta feature is the combination of multiple genes. Where is this? Oh, sorry, I'm going too fast. Okay, this is meta feature. These meta features will be part of your spatial enrichment column. But then we can plot these meta features. And so these meta genes are, in this case, are meta features that represent a bunch of very similar spatial expressed genes. And that really gives you some really cool patterns where you can see this, like this meta gene 1 and 2 is very distinct from each other. And it's a combination of multiple genes that show very similar patterns. Same for 3 and 4. You can also do it for more, and you can all of a sudden you can start seeing all these like beautiful patterns in the small piece of tissue. And then obviously one question you can do, which we don't really do, but it's kind of like straightforward, is you can do gene set enrichment, right, on these spatial co-expression modules. And I try to figure out like what exactly are these genes and why do they have this specific expression pattern. You can identify cell types and try to see, is it related to cell types? Is it related to some specific spatial organization? Whatever you can think of, it's your imagination. All right, so that's that. I think I want to show this thing real quickly. I kind of like to do this for any projects that we're working with. It's just to get, so once you have that set of genes, that spatial co-expression object, you can get a balanced spatial co-expression feature. So that simply extracts a similar set of genes, and you can do it in different ways from each of these spatial co-expression groups. And in this case, I'm extracting 5 genes from each of these co-expression modules. I give it the same name, color. I give it actually the same color for each, and then I can visualize this. And so what you see here, and I'm going to explain it here, is I think it's really beautiful when you do it this way. So all the colors are genes from the same spatial co-expression groups. So the green ones are 5 genes from that spatial co-expression group that had this very specific pattern. Same for red. And this is one way that you can create some really beautiful plots with genes representing different structures. So we're not really doing any type of biological assignment or annotation right now, but just visually you can start to appreciate how this tissue is being organized. All right, so last, the hexbin 25. So hexbin 25 is another factor 4, where we're going to even like make our data set more higher resolution. And so I would say almost unfortunately, I'm going to have to use a subset. I don't have to do it for my specific machine, but if you want to follow along on your local machine and you have a simple personal computer, you might run into issues if you try doing this on everything. So we're going to subset our Giotto object, and I'm kind of like visualizing which part I subset. So I can basically simply set the X min, X max, Y min, Y max, and that just creates this boundary box, and I can subset this out. That's what I'm showing here. So I can show you how it looks like all right, so this is that I was showing before. I've specifically said, hey, I want to show what the hex bin 100 looks like. This is our previous, we haven't created the hex bin 25 yet, but this is basically how our previous data looked like. And I say, hey, this is not at a high enough resolution, so I want to create a hex bin 25. This is the actual the actual transcripts in this subsetted area. Okay, so now I'm going to create my hex bin 25 in the same way as I did for hex bin 100. So we're going to tessellate. We're going to create new tessellations, so new hexagons, using a shape of 25. So that's the distance, the inner distance in the hexagon. We're going to set the polygon information. We're going to add the centroid locations, and we're going to visualize it again. And you will have to, you kind of like have to get really close to your screen and look at it, but now you have like really almost like very tiny hexagons, and you can start doing the same analysis that you've done before. And I'm running a little bit out of time right now, but you can do, for example, we're going to do again the overlap between the small hexagons and transcripts, overlap to matrix. We're going to filter it. We're going to look at everything. We're going to add the statistics. You can look at the feature data. I'm going to see if I can draw this up, the feature data. So this is the feature data. I just want to see if I can show you, showing this side by side. So in the smaller one, we don't have the same genes here, so it's a little bit harder to see. Anyway, you can look at it yourself. You can look at the feature data, P data, and just to kind of like get an idea what the mean expression was, or the total expression per. I should have actually looked at the cell metadata between the hex 400 and the hex 25, and all of a sudden you will see that with hex 25, oftentimes you only start capturing maybe like 50, 100 genes, and that gives you an idea like maybe you're already at a very low resolution and it doesn't really make sense to go any lower. At some point, if you go too low and you only detect like 10 or 15 transcripts, unique transcripts, then what are you really looking at is getting really sparse, and it's not going to be very easy to really understand what's happening. One thing I would like to show you very quickly is that you can, with the hex 25, let's say you did it on your whole tissue and it's getting really big and some of your functions are taking a really long time, you can use some of our projection functions. And what our projection functions try to do, and I'm going to show you one for PCA, UMAP, and the clustering results, is you're going to do it on a small subset of your data, and then you're going to project it on everything else. And it actually works pretty well, I would say, to some extent. Maybe not that well with this small data set, but if you do it with a really big data set where a subset is still a sizable portion of your data set, it's actually going to work really well. So we're going to identify the top, like the 25%, or what is the number of 25% of the total number of your. So in this case, we have 8,114 hexagons. And we're going to select 2028. So that's 25% of all the hexagons. And now we can say run PCA, but run PCA projection. And the only thing where it really differs is where I'm going to give it a different name just to make sure that I know it's a projection PCA, but you need to give it this random subset. And so it's going to run PCA on the smaller subset. And then it's actually going to, the next step is going to project it on the large data set. And by doing that, that actually works really well. And a very similar thing you can do with UMAP. With UMAP, it's also like, it's called run UMAP projection. It's the same as run UMAP, except that you run it on the random subset first, and then you project everything in the whole thing. And then you can actually see you have this one is the UMAP. One thing I forgot to mention, we have the PCA projection. We do have run PCA projection batch, which if you are really kind of like a little bit more anal about it, you can do it in batches and do it multiple times and combine all the data so that you have a little more robust output. So that's one way that you can speed up the PCA and the UMAP. Clustering with projection is similar in idea, but slightly different. here we're going to first subsample the Giotto object. So we're going to actively make a smaller object. We're going to perform clustering on the small Giotto object. And then we're going to project the cluster data from the small object to the full Giotto object using a very simple k-nearest voting approach, where you're going to identify your nearest neighbors in the full data set and how they are similar to the small data set. So here we create a subset again, the subset of hexagon IDs. We create a temporary Giotto object. We're going to use that temporary Giotto object and do hierarchical clustering, which I'm a big fan of. It actually works really well. It's just not really scalable, but you can do it on a smaller data set. And here I'm showing this. This is this like hierarchical clustering, which we have plotted over top of the UMAP. Hierarchical clustering doesn't always overlap with the UMAP because the UMAP itself is more based on the PCA space, and to some extent it does, but it's not perfect. But we can project these clusters. So what we do is we have our target object, that's our full Visium object, and then we have the temporary object. We can cluster it back, we can visualize it. Now what you see here is you get all your data back, including all the individual spatial hexagons. So we can visualize this. And this is what we get right now. So we have identified the cluster IDs for each individual hexagon on this like super low resolution. It doesn't really look great, but that's also probably for two main reasons. One, we actually removed quite a few genes, so we have a kind of like a very sparse data set, which is not great. But also sometimes when you have a sparse data set, your individual clustering results don't look really great. But if you want to have an idea about local tissue organization, then one thing you can do is doing niche clustering, and this is the last thing I will discuss. So niche clustering is where each cell will be clustered based on its local neighborhood. And I'm just going to run it and show it. So you also need a spatial network. That spatial network will determine what you consider as your local neighborhood. I'm considering 6 nearest spatial neighbors. You do a couple of calculations and then you add that back, and then you should see something like this. And so now all of a sudden you do see a lot more structure in the data, where you see like very specific niche locations and local domains and how they're organized in more like organized ways rather than the little bit more random organization of the individual cell types. And that's also probably how the actual data looks like. All right, so with that, I'm going to give it to Eddie for a few more minutes where we're going to discuss a little bit the database backend. So I did show you a little bit how we can alleviate some of the problems with big data, but there are still some memory problems with data ingestion, spatial operations, matrix operations, and also like the size of this matrix in spatial. And I think Eddie has some cool slides to show how we can alleviate some of that. Thank you. All right, Eddie, you want to start sharing your slides?
Yes, I think I need to become a co-host though to share my screen.
Okay. Yeah, go ahead.
Okay, great. Thanks, Ruben. So yeah, I'm just going to briefly go through some slides on what I've been working on recently to try and get at some of these problems that Ruben had mentioned regarding the scalability of these operations. And this has been an ongoing project of mine with George for the past year and a half or so. And basically, we've landed on using a database called DuckDB, which you guys may have heard about before. It's very popular right now in like the analytics field. But here's just a kind of visual representation in my head of like how DuckDB works in terms of its columnar format and why it's advantageous for doing things in our analytical type of workloads that often work on subsets of columns as opposed to entire groups of data, which is what I'm showing here. And it's depicted here in this GIF representing more of a row-based transactional database that a lot of people use for like your Amazon shopping cart type of situation. And so DuckDB is an embedded analytical database or an OLAP database that takes in things in a column-oriented fashion And this leads to a lot of performance improvements in terms of how the data is represented, which I'm trying to show here. In row base, you have a collection of heterogeneous types of data that often you don't need, and so that leads to kind of unnecessary space and data processing. If you compare that to the columnar format, you are able to compress data that's all in the same type because it's in the same column. This is easier to cache, it's easier to vectorize, and DuckDB has a very famous vectorized processing model that is very fast. And it's also easy to kind of take this data and work with it back and forth from the web to kind of like even like your Android phone. I think they're working on things like that. So it's really exciting, and we've been trying to figure out how to use this in Giotto Suite. And so one of the core problem we're trying to face is that a lot of laptops only have like 16 gigabytes of RAM or memory, but we have very fast solid state drives on like MacBooks. And so these are also significantly larger. And so if we can use this space in addition to the optimizations from DuckDB, perhaps we can address some of the bottlenecks related to these larger data sets. And so what we've done is we've actually gone ahead and made a few different packages that I'm going to go into briefly, called DB Matrix, DB Spatial, and DB Data, that wrap these various core operations that we are already familiar with in the R space for like matrix arithmetic and spatial operations in a database that is more scalable. And so this database operates through SQL queries that are recognized through DuckDB and can also be converted into Arrow. And so this is kind of the general approach that we're taking to try and address some of the scalability issues that we're facing with Giotto. Here's just an overview of the three packages that I just mentioned. The big idea is that we categorize the different data types into separate packages. In this case, DB Matrix is meant to handle a variety of numerical input data, so like CSVs, TSVs, matrix files, which have been mentioned previously in the Visium and Visium HD tutorial, as well as Parquet files. We also want to be able to work with in-memory representations of matrices that we are already familiar with through the matrix, capital M, and data frame packages. Bring those into DB Matrix, and we have our own like internal representation of these matrices that is compatible with DuckDB. It's essentially a triplet vector representation, and we're building out a whole bunch of generics that are based off of our familiar matrix package methods that we use all the time, like row, call names, dim, transpose, and a whole bunch of other statistical methods that you would expect to be able to run on matrices. We're also really interested, obviously, in doing things with spatial data. So how do we actually work with the largest spatial geospatial files that are out there? And so what we've also been working on is a package called DB Spatial that also is able to read in a variety of geospatial file formats, as well as our spatial objects from SF and Terra, and construct database representations of these based largely off of the DuckDB spatial extension. And these two core libraries are connected to a database on our local computer that is kind of organized at the lower level by DB data with like convenience functions and also caching the connection that a user might make to this database. And so here's just some preliminary benchmarks of DB Matrix with Giotto Suite. And we're comparing DB Matrix here in red to HDF5 Matrix, which is based off the delayed array framework, which we also have implemented in Giotto Suite. as well as an in-memory sparse matrix here in blue. And on the left, I'm just showing the runtime to go through our normalization procedure with these three different matrix inputs. And we can see here that we're not quite as fast as DGC matrix, which is still, um, kind of the fastest when you have the ability to actually work on in-memory matrices, but we're significantly faster compared to HDF5 matrix. And on the right here, we're comparing just the memory allocation. And generally speaking, we've seen that DuckDB or DB matrix is able to scale significantly better than HDF5 matrix and DGC matrix when it comes to the amount of memory that's used. So these were just for kind of smaller matrices. So this was 10,000 on the left here for runtime. And I think that's, what is it? 1, 2, 3, 4, 5, 100,000 for the memory allocation. We've also gone through and I've been currently battle testing DB matrix with like really large matrices. This is 50 million. DGC matrix isn't able to handle this data on my MacBook Pro with 16 gigs of memory. HDF5 matrix is, but we have been able to observe substantial speedups in terms of like on the order of like a magnitude and substantial improvements in terms of the amount of memory that's used as well. What's also great about DB matrix is that it adopts a very familiar syntax through the tidyverse's dbplyr and dplyr framework. And so if you are familiar with operating on data frames, if you've learned tidyverse, DB matrix and DB spatial are basically like a shoe-in to get working on as well. So I've also gone ahead and conducted some preliminary benchmarks of DB Spatial, which again is our kind of scalable approach to doing operations on spatial data in a DuckDB database within Giotto Suite. Here I'm just showing three different regions of interest of increasing size that have increasing number of cell polygons. And what we're doing is we're just doing kind of a simulation on intersecting polygons that were generated from different segmentation algorithms for the same number of cells for each of these ROIs. And we're comparing how much time it takes to find the intersect between these polygons as we increase the number of cells across these three ROIs. And we can see that DB Spatial is significantly faster than the equivalent intersection method in the RSpatial SF package, which is doing things in memory up to like a magnitude performance improvement. So this is just a preview of what we've been working on. We're very excited to be at the DuckDB conference next week, where I'll be talking more about these packages. And also we are actively working on wrapping these packages in Giotto DB so that we can address a lot of our scalability issues. But we also understand that these issues are applicable to a lot of people, not just those running Giotto. So we're trying to make sure that these packages are generalizable to other types of applications as well. So really excited to be able to release the code soon. And the plan was to release it today, but unfortunately we aren't quite there yet, but still working towards it. So